away from the center of the bony shaft as the latter lengthens. So they have the EPP shell. The parts of the EPP shell are the area where cartilage is being laid down by chondrocytes, lines, while the EPP shell growth plate is the line. The shaft edge where osteoblast converts cartilaginous tissue to bone. As long as the epiphyseal growth plate exists, linear growth of the shaft can take place. However, it ceases when the plates are themselves converted to bone as a result of hormonal influence at puberty. This is known as epiphyseal closure and occurs at different times in different bones. Therefore, a person's bone age can be determined by X-raying the bones and determining which one have undergone epiphyseal closure. An important factor here is that bone is constantly being remodeled by the osteoblasts and osteoclasts working together. The purpose of remodeling is to regulate calcium homeostasis, repair micro-damaged bone from everyday stress, but also to shape and sculpture the skeleton during growth. Osteoclast reabsorb old bone and then osteoblast moves into the area and lay down new matrix which become calcified. This process is dependent in part on the stresses imposed on the bones by gravity and muscle tension, both of which stimulates osteoblastics activity. When osteoblasts are stimulated, there is an increase in the bone mass through increased secretion of osteoids and by inhibiting the ability of osteoclasts to break down osseous tissues. Bone building through increased secretion of osteoid is stimulated by the secretion of growth hormone by the pituitary, thyroid hormone, and the sex hormone or the estrogen and androgen. It is also influenced by many other hormones as summarized in the table below. Hormones that favor bone formation and increase bone mass are insulin, growth hormone, insulin-like growth factor 1, estrogen, testosterone, 1,2,25-dihydroxy, vitamin D3, which influence only mineralization, not matrix, and calcitonin. Bones that favor increased bone reabsorption and decreased bone mass are the parathyroid hormone, cortisol, thyroid hormone, the T4 and the T3, and that's there. So let's talk more about the growth hormone. Growth hormone causes growth of the epiphyseal region of the long bones. Growth of the long bones can be monitored by measuring the incorporation of sulfur into the epiphyseal cartilage. It is said that growth hormone acts indirectly on bone by ways of the production of a sulfation factor. The sulfation factor is known to consist of several peptides referred to as somatomedins. Injected radio-labeled growth hormone rapidly localized to the liver rather than to the epiphysis of the long bones. Somatomedin is generally used to refer to those growth factors found in the plasma that are under the control of growth hormone, have insulin-like properties, and promote the incorporation of sulfate into cartilage, the somatomedin hypothesis. Insulin-like growth factor 1 and 2 or IGF-1 and 2 are two substances isolated from the plasma in pure or rather pure form fulfill this criteria. The peptides bear some sort of structural relationship to proinsulin and therefore exhibit some affinity for insulin receptor. Growth hormone does not have a direct effect on cartilage, but rather stimulates chondrogenesis and subsequent growth indirectly by ways of somatomedins. According to the somatomedins hypothesis, the number of IGF-1 immunoreactive cells in the proliferative zone is increased. IGF-1 is produced in the proliferative chondrocytes in the growth plate in response to growth hormone. Growth hormone can induce local IGF-1 production in the epiphyseal plate at the level of both mRNA and protein. Growth hormone but not IGF-1 stimulates the multiplication of the slow-cycling label-retaining cell in the germinal layer of the epiphyseal plate. Fat is located in fused IGF-1 is able to increase epiphyseal width as well as longitudinal bone growth. Children manifest two periods of rapid increase in height. during the first 2 years of half-life and during puberty. Note that increase in height is not necessarily correlated with the rates of growth of specific organs. The pubertal growth spurts last several years in both sexes, but both during this period is greater in boys. This, plus the fact that boys grow more before puberty because they begin puberty approximately 2 years later than girls. Account for the different storage height between men and women. Graph below show the relative growth in the brain and total body height and reproductive organ, which the brain is higher than, followed by total body height, and then the reproductive organs comes in. Now let's talk about the environmental factor influencing growth. The primary factor influencing growth are the adequacy of nutrient supply, freedom of diseases, lack of sufficient amount of any of the essential amino acid, essentially fatty acid, vitamins, or minerals interferes with growth. Total protein and sufficient nutrients needed to provide energy must also be adequate. The growth-inhibiting effects of malnutrition can be seen at any time of development but are most profound when they occur very early in life. Thus, maternal malnutrition may cause growth retardation in the fetus. Since low birth weight is strongly associated with increased infant mortality, prenatal malnutrition causes increased numbers of prenatal and early postnatal deaths. Moreover, irreversible stunting of brain development may be caused by prenatal malnutrition. During infancy and childhood, too, malnutrition can interfere with both intellectual development and total body growth. Following a temporary period of stunted growth due to malnutrition or illness, and given proper nutrition and recovery from illness, a child manifests a remarkable growth spurt, catch-up growth that brings the child up to the normal height expected for his or her age. The mechanism that accounts for this accelerated growth is, however, unknown. Hormonal influence on growth. Human growth requires hormones. A hormone is chemical released by a cell or a gland in one part of the body that sends out messages that affects particular cells in other parts of the organism. Only a small amount of hormone is required to alter cell metabolism. Okay, the most important hormone to human growth are the growth hormone, the insulin-like growth factor 1 and 2, the thyroid hormones, testosterone, estrogens. The site of hormone production is due in the hypothalamus, in the pituitary gland, the thyroid, the thymus, the adrenal gland, the pancreas, ovary, and testicles. There is also a large group of peptide growth factors, which includes the insulin-like growth factors. And most of these growth factors act as paracrine and autocrine agents. Paracrine is a chemical signal that diffuses into the area and interact with receptors on nearby cells, the release of neurotransmitter and synapses in the nervous system. Autocrine, on the other hand, is the cell signals itself through a chemical that it synthesizes and then responds to. Autocrine signaling can occur solely within the cytoplasm of the cell or by a secreted chemical interacting with receptors on the surface of the same cell's agent. This type of hormone stimulates the differentiation and or sometimes cell division of specific cells. Generally, the term used for a chemical that stimulates cell division is called a mitogen. Growth is also modulated by peptide growth inhibiting factors which inhibit cell division in specific tissues of the body. So, insulin-like growth factor 1 and 2. Growth hormone exerts its cell division stimulating effect not directly on cells but rather indirectly through the mediation of a mitogen whose synthesis and release are induced by growth hormone. This mitogen is called insulin-like growth factor 1, IGF-1. Under the influence of growth hormone, IGF-1 is secreted by the liver, enters the blood, and functions as a hormone. The importance of IGF-1 in mediating the major growth-promoting effects of growth hormone is shown by the fact that dwarfism cannot be due to only to decreased secretion of growth hormone but also to decreased production of IGF-1 or even failure of the tissue to respond to IGF-1. IGF-1 is required for normal fetal total body growth and specifically for normal maturation of the fetal nervous system. The stimulus for IGF-1 secretion during prenatal life is however unknown at this time. Finally, it should be noted that there is another messenger, insulin-like growth factor 2, that is closely related to IGF-1. IGF-2, the secretion of which is independent of growth hormone, is also a crucial mitogen during the prenatal period. It continues to be secreted throughout life, but its postnatal function is unknown. The growth hormone is secreted by the anterior pituitary gland. We're going to talk about the growth hormone. So the growth hormone is secreted by the anterior pituitary gland. It has little or no effect on fetal growth. However, it is the most important hormone for postnatal growth. Main growth-promoting effects is the stimulation of cell division in many particular tissue regions. Growth hormones promotes bone lengthening by stimulating maturation and cell division of the chondrocytes in the epiphyseal plates and thereby continuously widens the plate in providing more cartilage for formation of bone. Chondrocytes are cells found in the cartilage. They produce and maintain the cartilaginous matrix, which consists mainly of collagen and proteoglycan. So, pituitary gland secretes growth hormone, which goes to the muscle growth, bone growth, IGF-1, muscle growth, then the growth hormone goes to the liver, then IGF-1 do the rest as the bone growth. So, what are the thyroid hormones? The thyroid hormones include thyroxine, which is secreted by the follicular cell of the thyroid gland. Triiodothyronine is released from the pituitary gland. It affects almost every physiological process in the body, including growth and development, metabolism, body temperature, and heart rate. Both are essential for normal growth because they are required for both the synthesis of growth hormone and the growth-promoting effects of the hormone. Infants and children who are deficient in thyroid production usually show signs of retarded growth due to the slow formation of bone growth. This deficiency is termed as hypothyroidism. Thyroid hormones are also essential for normal development of the central nervous system during fetal life. Inadequate production of maternal and fetal thyroid hormones due to severe iodine deficiency during pregnancy is one of the most occurable instances, yet still the most common preventable causes of mental retardation. This is termed endemic cretinism. This effect on the brain development must be distinguished from other effects. Thyroid hormone exerts on the nervous system throughout the human life and not just during infancy. Therefore, a hypothyroid or undersecretion of thyroid person will exhibit sluggish reaction and poor mental functions. However, these effects are completely reversible at times with administration of thyroid hormones. So, to a person with hyperthyroidism, excess of secretion of thyroid shows signs of being jittery and hyperactive. So, thyroid system. So, this is the hypothalamus, which the thyrotropin releasing hormones goes to the thyroid stimulating hormones and goes to the gland, which the thyroid glands, it's either negative, if it is in the negative feedback, it goes to the anterior pituitary gland. If it is not, then thyroid hormones, the triiodothyronine and thyroxine, increases the metabolism, growth and development, and increase catecholamines effects. Insulin. Let's talk about the insulin. Insulin is a hormone central to regulating carbohydrates and fat metabolism in the body. It causes cells in the liver, muscles, and fat tissues to take up glucose from the blood and store it as a glycogen in the liver and muscles. Therefore, it is obvious that an adequate amount of